Alrighty, so for science, we're looking at mutations. Thanks to Sam Moyle for producing this PowerPoint. So the science understanding we're looking at, changes in DNA sequence are called mutations. Mutations in genes and chromosomes can result in errors in DNA replication or cell division, or from damage by physical and chemical factors in the environment. And mutation rate can be increased by ionizing radiation, mutagenic chemicals, and viruses. Compare the potential consequences of mutations in germ cells and somatic cells, and explain how inheritable mutations can lead to changes in the characteristics of descendants. So mutations are changes in the genetic sequence. This happens relatively often, and it's very important because in the end it can drive evolution. They can occur spontaneously throughout your life, but they can also be induced, and we'll look at some factors that can induce mutations later. Any change in the DNA sequence can have a big flow-on effect, so this can lead to 3 to 1. Any change in the DNA sequence can have a big flow-on effect, and this can lead to a change in the proteins that are being produced. And those changes in the gene products can lead to all kinds of disorders. So examples, here we have some extra legs and things being produced. Down here we have some extra legs being produced. Sickle cell anemia is a blood disorder. Tay-Sachs disease is a very nasty genetic disorder. Cystic fibrosis is reasonably common genetic disorder. So cystic fibrosis is a protein that produces a channel that moves chloride ions in and out of cells. That channel isn't shaped quite correctly, and that has huge impacts in terms of health. So let's look at some different possible types of mutations. So missense mutation, this is where, here's our regular DNA sequence. Here we're replacing a single A in the sequence with a C. By doing that, we change the DNA triplet, and that produces a different amino acid once a translation has occurred. This could be bad. The protein might not fold the right way, and then the entire protein might not do its job correctly. It could be good. It could be an advantage. The protein might work better. Or it could make no difference at all. So there are some possible outcomes of mutations. A nonsense mutation is where, again, we're changing a nucleotide. So we're changing, in this case, a C to a T. But that change in nucleotide changes the DNA triplet, and that changes the outcome in the gene product at the end. So rather than producing glycine, we produce a stop. So that means everything that follows after here isn't produced, and that can mean the entire protein might not work. Here's an insertion. This is where you inserted a nucleotide. So we were going TCAT, now we're going TACAT. So we've introduced an A. Introducing the A will change all the triplets afterwards, and therefore all the codons and anticodons, and eventually the amino acids that are put into your sequence. So that can be quite deleterious, that can be quite nasty. 3, 2, 1. The opposite of an insertion is a deletion. So this is where a nucleotide gets deleted. So here's our regular sequence again, so CAT, and then after our mutation we have a CTC, so we've gotten rid of that A. And again, that's changing the amino acids that are being produced from this DNA sequence. These are just some examples of mutations. There's many other types of mutations that can occur. So some causes of these mutations can be from just DNA replication and cell division. Others can be caused from outside sources. And let's have a look at some of those now. So mutations can be induced by environmental factors. So ionizing radiations, this is high energy radiation. So X-rays, you need to wear a lead belt whenever you're getting X-rayed. UV light, so skin cancer in Australia, this is a very common cause of skin cancer. Mutagenic chemicals, so in cigarette smoke there are things like 3 to 1. So mutagenic chemicals, so in cigarette smoke there's many different chemicals that can lead to changes in the DNA sequence, and this is one reason why cigarette smoke causes large amounts of lung cancer. And then some viruses can lead to changes in the DNA sequence. That's what they do. Viruses, they change DNA sequences. So hepatitis B and C, for example, are very commonly related to liver cancer. Usually these mutations that are induced by environmental factors, they only affect somatic cells. So somatic cells are just your regular, everyday body cells. So they'll only affect the individual organism. If you get mutations in your germ cells, these are in your sperm or your eggs, those mutations can be passed on to generations after you. So germ cell mutations affect the zygote, and therefore that will occur in every cell's DNA in that zygote in the new organism being produced. So mutations can be inherited from one generation to the next. This is one reason why when getting X-rayed, they're very careful with females to protect the ovaries when X-rays are occurring. ovaries are where the eggs are stored. So if you're zapping them with large amounts of X-rays, you could change the genetic sequence of those eggs, and those that could lead to mutations being passed on into offspring. So some diseases have a genetic basis, meaning they can be contracted via inheritance from parents, and then grandparents, grandparents, and so on. Now there's a Mendelian aspect to a lot of this. So Harlequin's disease, for example, which is quite a nasty disorder, there's always that element of chance. So it's a recessive disorder. So if your parents have one copy each, then there's a 1 in 4 chance of that being produced. Other examples, phenylketonuria, thalassemia. So this is where you can't absorb phenylalanine in your diet. That leads to blood disorder. Sickle cell anemia, which we talked about before, is a blood disorder, and also haemophilia, which leads to platelets not working particularly well. Now, generally speaking, in terms of inherited disease, you need 2 copies of the allele of the affected gene. So it needs to be a homozygous disorder in order for it to be expressed in the phenotype. But then there are sex-linked disorders linked to the X chromosome. So in sex-linked disorders, often males will only express the disorder since they get 1 copy of the faulty X chromosome. They don't have a backup X chromosome since their other chromosome they have is a Y chromosome. However, females that get 2 copies of the faulty X chromosome can also express the phenotype. And we'll look at haemophilia now. So this is Queen Victoria and Albert's legacy of haemophilia in royal families all throughout Europe. So Victoria got a spontaneous mutation, although there's questions about whether it was a spontaneous mutation or not in terms of her parentage. So she was carrying 1 faulty X chromosome, so 1 chromosome that's carrying the haemophilia gene. Her husband was regular, Albert. They had many kids, and from those children we have, say, Alice over here, who's carrying 1 X with the haemophilia gene. We have her son Leopold. Again, he had 1 X, but he didn't have a backup X as if he was a female. He had 1 X and 1 Y, so Leopold had haemophilia. And then we have Beatrice over here, who was also a carrier. And we can see that they went through the royal families. There's lots of intermarriage between royal families in Europe, and these genes were being passed on all the way through. Now, if we have a look here, this is the Russian royal family down here. So this is Tsar Nicholas II of Russia. His wife is descended from Queen Victoria. She was carrying 1 of the X chromosomes that had the haemophilia gene on it, and they had a son called Alexei who had haemophilia. Now, some argument that one of the causes of the Russian Revolution was due to Tsar Nicholas II caring more about looking after his sick son with haemophilia than looking after managing the state of Russia, and that eventually led to the Russian Revolution. So there's one argument made that this 1 faulty gene up here led to the Russian Revolution and the spread of communism in Russia. Now, mutations aren't necessarily bad, because mutations and changes in genes are what eventually leads to evolution. The reason for that is differences in genotype leads to differences in phenotype, and some phenotypes are more successful in the environment than others. When we're talking success, we're talking about having babies. So if a change in a phenotype due to a change in a genotype makes that organism more likely to survive and reproduce and pass on their genes to the offspring, that can lead to big changes over time, and that's what evolution is. So mutations aren't necessarily a bad thing. That's it for Science Today. Bye.